Hi, my name is Julieta Flores, and today I will be talking about my study, Effects of Body Size and Group Size on Exploratory Behavior in Hermit Crabs. First, we're going to talk about the relationship between behavior and size. A study showed that the ability to detect the influence of boldness and body size on fish behavior in groups was weaker in more sociable individuals. The same study showed that individual boldness on whether a fish initiated the first move is affected by body fish length. Another study showed hermit crab personality can affect individual fitness and the dynamics of populations. Next, I looked at social interactions as it relates to size. The previous study showed dependency on individuals' sociability suggests that the degree of sociability will have an indirect effect on behaviors that are found to correlate when individuals are alone or in groups. Additionally, boldness and sociability were not found to be correlated in non-social animals. However, hermit crabs' personality can affect individual fitness and dynamics in population. Crabs emerged faster in groups of 10 and 20 than they did in groups of 2. Observations. Exploratory behavior in hermit crabs may vary with individual body size and social groups. My hypotheses were that crabs would be more active in social groups. Smaller individuals will show greater activity in larger groups. That size will affect activity, meaning larger individuals will be more active compared to smaller individuals. Lastly, taking from the study previously mentioned, boldness and sociability were not found to be correlated in non-social animals. However, hermit crabs are known to be social. Trial methodology. Hermit crabs were split up into groups of 3 or 4 and then groups of pairs. Within each group, the hermit crab was labeled as largest, intermediate, or smallest, depending on the weight of each crab. The time was kept for showing boldness / personality, which meant time spent outside of its shell, as can be seen in the top picture. This included walking around, attempting to escape, and just standing outside of its shell. No boldness shown is the bottom picture, stasis inside the shell. Food cycling. Crabs were fed and trials were ran at night when most activity occurred. Food cycling was done to ensure that crabs would be active during the night of test trials. The entire group was fed at night and the food was removed in the morning. There was 1 day of starvation, meaning there was no food given to them at night, and the test trials were done the next day. This ensured that the test trials were done when crabs were naturally most active and gave motive to the crabs. Night of test trials. On the night of test trials, all crabs were gathered before the test trials began. They were placed within a bowl inside of their tank. Hermit crabs were randomly picked for their groups and pairings. Time trials lasted 15 minutes, and the food selected was granola and crushed hermit crab pellets. All test trials were made in a separate, smaller tank where they can be more easily observed. Now we're going to be talking about what we found. So when we looked at the time spent outside of the shell versus the size of each hermit crab by groups and pairs, we saw that intermediate crabs spent the most time outside of the shells. And when in pairs, crabs spent nearly the same amount of time outside of the shell, no matter the size. Time spent inside the shell versus size by groups and pairs, intermediate crabs spent the least amount of time in their shell. The largest crabs, when in groups, spent the most time in the shell. And the largest and smallest crabs spent nearly the same amount of time in their shell when in groups. There is a small difference between the largest and smallest crabs in pairs, but it was not significant. Successful escapes. Crabs in pairs never successfully escaped. Crabs that successfully escaped were not always the largest in their groups, as seen in this graph. Observational data. This was data noted during test trials. Smaller crabs flinched when approached by larger crabs. Larger crabs would use smaller crabs to crawl on top to help them escape. And larger crabs more often were the first to start exploring. No statistical data or numerical data was taken for these observations. Discussion. These statistics took into consideration the largest, smallest, and intermediate-sized crabs in both groups and pairs. So when looking at stasis inside of a shell, the largest crabs had a z-score of 0.1486. Inside of the shell, for the smallest crabs had a z-score of 0.5186. Outside of the shell, the largest individuals had a z-score of 0.5169. Outside of the shell, for individuals that are the smallest, had a z-score of 0.3662. Intermediate-sized crabs were not giving z-score values. The reasoning for these z-score values is because of the small number of test trials. To get a more accurate representation, I would have had to had more test trials done. In conclusion, smaller individuals will show greater activity in larger groups. However, my study showed that smaller crabs were more active in pairs versus groups. For the hypothesis that larger individuals will be more active compared to smaller individuals, there is no significant difference. When it came to the hypothesis of boldness and sociability not correlated in non-social animals, when considering hermit crabs are social animals, there was still no correlation found. Overall, there was no significant difference between crab sizes in any case. Ultimately, there were not enough trials ran to give definitive answers. These are my citations, and I thank you for your time.